Dr. Ramy, can you hear me now?
Yeah, Dr. George is here now. Yeah,
Okay.
Thank you. Technology
Okay.
is always surprising. So Dr.
Yes.
George Maher is the CEO, the head of MARC Research Center. He will welcome us and give us a summary of what we do here at MARC.
Okay. Thank you, Dr. Ramy, and thank you for all the attendees. Thank you for Dr. Mohamed Khayyal for being with us, and thanks for Alex Mira for joining us today. I will be very brief in giving shedding some light on MARC. We are a clinical and medical research center that have a vision of improving lives through translating basic research into medical solutions and healthcare solutions that improve lives. In MARC, we believe that part of our philosophy is to support research and researchers through many different activities, through offering some services and through giving some grants to young researchers, and also through organizing such meetings like our today webinar. We have a series of webinars named Remedy Within. Our tonight webinar is part of our long-term vision in supporting research and researchers. I will not be much detailing about MARC more than this, and I'm leaving the floor to my colleague, Dr. Ramy, the head of Bioscience Labs in MARC. Dr. Ramy, floor is yours.
Thank you so much, Dr. George. It's a great pleasure actually for me to be at MARC. I'm a new member, relatively less than a year now, and I'm still exploring the many ways we can try to bridge, which is the main mission of MARC, bridging industry, the huge industry with the huge academia, academia always does research and says we want to bridge the gap. However, the initiative really always comes from, it's better to come from industry because it happens in many places around the world. And our guest of honor today has a great and deep view about that, about how industry can support research and how can this research be translated to the benefit of humanity and to also the economic benefit of the country. And so our honorary speaker and guest today, our honored guest is Professor Mohamed Khayyal, whom I will introduce in detail. Dr. Riyad regrets very much not being able to attend. He was looking forward to this very much, and unfortunately he has a board meeting that probably extended more than it should. And Dr. Khayyal is a hero of all of us who has been at Faculty of Pharmacy, Cairo University, as well as many pharmacologists around the world, of course. And he believes very much in the link between industry and research. Our other guest is Dr. Alex Mira from Valencia in Spain, who has a very wonderful story. He will tell us later again about how he is academic completely, but he also translated his research into industry. So for Dr. Khayyal, I don't know where to start. Should I start with, I don't know, 1992 when I attended my first pharmacology lecture, and I was so surprised. I'm not known among different people and among my friends for being, exaggerating or anything, but I was really impressed in that lecture because I was listening to I said it. I even didn't know what it meant then. I was a second-year student, but I said, this sounds like a seminar, a scientific talk. The speaker, Dr. Khayyal, was speaking perfect English, fluent English. We had a book that we were studying from, not like these sheets of paper that we photocopy. And he was talking about epinephrine, norepinephrine, making us laugh. But I cannot also forget that I left the exam almost, instead of answering the question, I kept laughing because he always had a poem in his exams. And I try to do this with my students, but my poetry talents are very weak. However, he had always a poem, and unfortunately, I didn't discover until the year after that the poem had the key to solve the questions. I just thought it was for fun, but it had actually the key for the answer. So that's how I met Dr. Khayyal, and I knew him. And then later we discovered he's a great scholar and researcher. I don't know if I can also share some some slides. Mohamed, Sam, can I share the slides from here or?
Sure.
Okay,
Yes,
so
sure you
I
can.
have some slides. I hope I can be able to share. Just pictures from. Can you see the slides?
Yes, clear.
Of course, it's a courtesy of, through Dr. Khayyal, I could receive these slides from some presentation that was honoring him, and I will not take long time or anything. Again, he knows better about these slides than me, but I will try to show. So first of all, let me do the official bio, although it can take an entire hour. So I'll be again brief. So Professor Khayyal is currently an emeritus professor of pharmacology at Faculty of Pharmacy, Cairo University, which I am also honored to be a professor there academically. However, he has been a professor much longer than me. He has been also the chair of, and one of the co-founders of the department, but also the chair of the department for many years, the Department of Pharmacology. Then Professor Khayyal also was the vice dean, one of the first vice deans of Faculty of Pharmacy, Cairo University. But what he sent me as information goes way before that. So again, I'm not leaking any secrets, Professor Khayyal. This is what you sent me. So it starts here, and then with a family of pharmacologists. And he went to the Victoria College in Alexandria, and it's a British school, and that's now I know why he is so fluent in English. However, I don't know why he is also fluent in French and German, and also Arabic, of course, because many people don't speak their mother tongue. So Professor Khayyal is multilingual, polyglossal, right? Is this the right term? He was active since school at Boy Scouts. He went to Germany early in his life, but he went to get his PhD in London, and that's what he visited later. This is with his father and PhD. I believe Professor Gadam is your PhD mentor, right? And international relations in London, always active. His graduation, engagement, many recognition by the press and many things. And then he was around the world, China, Australia, Germany, conferences. I don't want to go far because we want to listen to Dr. Khayyal telling us about his how he sees his career in research, but a very, very, very achieved career. And he worked on remedies of one of the biggest problems in Egypt, which is schistosoma or bilharzia. And then now he's very, very interested in the metabolic disorders of the intestine and autoimmune disorders, IBD, and looking for herbal products, but standardizing them with animal experiments. He is also a person who is known for his sense of humor. I never talked to Dr. Khayyal without having him make a small pun, playing on words, and always making me laugh. Has always been loved by his students everywhere in the world. I don't want to take longer time because now the floor is yours, Professor Khayyal. We really want to honor you, but as I told you when we were preparing for this, he refused completely. He said, he said, I didn't do anything special. I told him, no, you inspired many generations.
Something went wrong.
Okay, no, I can hear you now.
Sorry,
we can hear you.
something went wrong. Just a minute.
No, we can perfectly hear you and see you.
I don't know what happened now.
Sorry?
Something went wrong somewhere.
But we can hear you and see you. Maybe you cannot see yourself, but we can. So we just wanted to know your opinion on the role of
I don't know. Okay, thank you so much. I feel very privileged to be with you today or tonight. I'd like just to tell you, when you chose the title of the webinar, Medicines from Within, actually, the remedy within is within here, not within the intestine. Because you have to really think before you leap. You have to think. And I remember quite distinctly when I first went to London to study for my PhD in the early 1960s, my professor sent me 1 year to spend 2 months with every one of the department to choose a to choose a topic on which to do my PhD. After spending 1 year from one lab to the other, he told me, look here, forget all what you've seen. When you do research, do something that will help your country. And I told him, well, I don't know. He told me, look here, your country is suffering a lot from bilharziasis, schistosomes. I had no idea about schistosomes. But he told me, look here, you will learn here about the worms. No one in the department knew anything about them. And yet he sent me to a department outside the School of Pharmacy where I started to know what a worm looks like, how to treat the worm, and I spent 4 years working on worms and on diseases of the bilharzia. And after finishing my PhD on schistosomiasis, I came back to Cairo and worked about 20 years on the worms. And then I found out that, well, the country has enough studies and there is no need to do any further work on this subject. So I started developing a new idea, and that was what was common between bilharzia and other inflammatory disorders was the intestinal inflammation. And I started moving on to herbs because I found out that many industry companies were interested in developing herbs. And I stayed with herbs ever since. Of course, I did a lot of work around the world with many, many other companies, not only from Germany, but from the States and Holland and Europe. And I'm very happy to have achieved a lot so far. And until today, I still love research work, but it has to do something with developing health and taking care of health problems around the world. It's not a question of doing research by adding A plus B and see what happens. This is not really research. Research work has to do with developing something that will improve humanity, it will improve health care, and this is something that is lacking nowadays in our way of thinking. Research means do something which will, there's a problem somewhere, and you have to resolve it in order to get somewhere to benefit mankind and to benefit healthcare. I'm not in favor of doing just research for the sake of research. And unfortunately, nowadays, the mentality of people, mainly in Egypt, to do research is to get a title. Title means nothing. Do research, you continue to do research, because you love research, irrespective of where it will get you. But don't do research work in order to achieve a title. And I wish this would be common nowadays more and more, because I have to make a little joke, because, you know, I always like to do jokes. And in companies all over the world, they have what we call R&D, research and development. But unfortunately, in Egypt, many companies have now converted R&D into R&D, which means nothing at all. I'm sorry for those who don't speak Arabic, but, you know, research means to do something which will help humanity, and not just do research to obtain a title. And I wish this would continue nowadays. In my department, at least, all those that have done research with me, they know that I don't choose any topic. except if it will help solve a problem somewhere. Thank you so much. I'm at your service, but I cannot praise myself, but I'm very happy to be here, and I feel very privileged to have been among you. And MARC in particular, I must say something about MARC, because Dr. Riyad and his late father were good friends of mine, and I admired the mentality of Riyad to build up MARC, because research here means something. And I hope it will continue that way, and it will propagate further companies to follow the same principles. I thank you very much for your invitation to me. I don't really believe that I did something, but I love to be down to earth. One day I will be below the earth, but until I'm below the earth, I love to be down to earth and never think highly of myself. I try, and I continue to try. Thank you so much.
Thank you so much, Professor. You always make us smile, but what you said is really, really very deep inside, because the quest for titles, you cannot have two masters, right? So if you look for a title, then you're not looking for science really, or for helping people. Well, I know that you said you don't want to talk much, but we want to hear one more thing from you. Well, it may be a stereotypic question, and but I'm looking at the positive side. What one thing that has changed during the past many years? I know that many things have changed negatively, but what is one positive thing that we can benefit from now, that was not there earlier, you know?
A very difficult question, because you have to change the mentality of those in charge. I'm happy to see that in MARC they have the right mentality, and this gives me a boost and optimism that they will continue to do so. And I'm happy to say that I have not seen that in other companies in Egypt. In Europe, yes. And I am a follower of R&D all over Europe, in Germany, in Holland, in Sweden, in England, in Australia. Yes, I've been everywhere, and I continue to do research with companies abroad. I'm not very keen on doing research with companies here because of that particular point. I want research for the sake of research and for the benefit of mankind. Thank you very much.
But we hope, since you have, we have a positive view of MARC, we hope that we can benefit from some collaboration with you, inshallah. And we hope we don't disappoint you. So Professor, can you say one last thing? How did you come up with all these poems in the exams? So now we can have a poem on the artificial intelligence on ChatGPT, but Professor Khayyal always used to have a song or a poem that can solve the whole exam.
Oh dear. It's my upbringing, you know, having been a British maid and all my life. I like to play on words because I have nothing else to play with. So words are fine. And it's just you have to be proficiency in the language. And alhamdulillah, I'm happy to say that my British education, I have an Oxford Cambridge school certificate, and well, I'm British, alhamdulillah, and this helps me to put words together, to design a poem. For me, it's easy. I'll develop something about this meeting tonight and send it to you later on. Okay.
Thank you so much, Professor Khayyal. Professor Khayyal is an emeritus professor at Faculty of Pharmacy, Cairo University. He's the the co-founder or the founder of the pharmacology department at Faculty of Pharmacy, Cairo University, but also he comes from a pharmacological family, and he's a founder and a pioneer of pharmacology around the world. Before ending this first part, Dr. George, do you want to comment or ask any questions to Dr. Khayyal before we go to the next speaker?
It's really our pleasure having Dr. Khayyal together with us today. And it's our honor if we can have any kind of collaboration with his kind side with MARC. And I hope, as you said, Dr. Ramy, we will not disappoint him at any time. Thanks, Dr. Khayyal, for being with us today. Thank you.
Thank you very much.
Thank you very much for being with us at your evening time and with all the technological challenges we had. There is one message.
One small thing, really, Ramy. Since you wanted me to play on words, I want to say something to Dr. Alex, because although I've been to Spain several times, and I studied even Spanish for 3 years in Cairo, but I had no chance to practice it very much. And I said, now, todos oblidad. I forgot everything, never mind. But what I found very nice about Spain, they have a very famous personality called Ramon Khayyal. Ramon y Khayyal. And I always thought Khayyal was like by Khayyal. But it was different. Ramon y Khayyal, and I'm Mohamed Khayyal, so there is something in common. Thank you so much.
Thank you so much, Professor. And there are 2 messages in the chat. So one is from Dr. Mohamed Abdul Hakim. He said he's very honored to listen again to Dr. Khayyal speaking after 27 years. Last time he was at Faculty of Pharmacy. He's now a professor in the US. There is also one from Mina Abadir. He said, it's honor to meet the distinguished Professor Khayyal. He worked in top 11 pharma companies from clinical track development, and he is very, very happy to meet you. He has a question, but maybe we can keep it for the discussion later about the pharma companies. So with this, I'm really, really grateful to Professor Khayyal, and I know that I burdened him a lot with phone calls this week to beg him to be with us, and I'm very happy he accepted our invitation. And now another movement to talk about the link between industry and academia, but also to talk about translating research. We chose many titles for the next talk. So one of them that passed through censorship was Can Marriage Save Your Teeth? From Keris, and a beautiful story of translating research. And this is by Dr. Alex Mira. Dr. Alex, you know, he has been to Egypt a couple of times. He knows that everybody loves Alexandria, and also we call Alexandria Alex. And Dr. Alex Mira has he has been around the world as well. He has gotten his PhD from Oxford University in the UK, but then moved to the US for postdoctoral research, as well as Sweden. He also worked in a great lab in the US, Pioneers of Genomics, the lab of Dennis Ockman, right, and Howard Ockman, sorry, and Nancy Moran, and then he made the choice that Professor Khayyal has made, and many of us, as he went back to his country, he went back to Spain to work in a laboratory of public health. It's called FISABIO. Maybe Alex can tell us what it stands for, but he has been since then interested in studying the human microbes and how they can actually improve our health. And Professor Alex Mira has, as I told you, has visited Egypt a couple of times, gave a talk in Alexandria, and also gave a talk in Cairo. And we always love to see him virtually or personally. And again, without taking more time from science, I will ask Dr. Alex Mira to tell us the story, as I asked him, not just the science, but the story, because it's very interesting. And I don't know, last time I heard from the story was 5 years ago, so I know that there are great I think, great steps ahead after the discovery. So please take the mic, and if you want to share your slides.
Okay, let's see if I can share my screen. Okay. Okay, I'll do entire screen just in case.
You can either share the screen or one window, whatever you like. Yeah.
I think I'll do the full screen just in case. Can you?
Yeah, we can see
it already? Okay.
Yes, yes.
Okay. Well, thank you very much for the invitation, and thank you, Ramy. Yeah, I mean, it's a big honor for me to be here as usual, and I miss you all very much. I hope we can meet in person again soon. And you have been wonderful hosts every time I've been in Egypt, and it's a wonderful country. And I realized how similar we are in character and in many things in life. So it has been a truly wonderful experience to get to know you. And well, I work at the FISABIO Institute in Valencia, Spain. You were asking, I mean, FISABIO stands for the Foundation for the Promotion of Biomedical Research. And it is a public research institute, so I'm very lucky to be able to live out of science. So I do some teaching, but mainly my job is mainly a full-time research. And the main line of research in my lab has been actually to look at not what happens to individuals with disease, but actually to look at what happens to those individuals that apparently appear to be immune to disease, in particular immune to infectious disease, because we believe that by understanding how health actually happens and how is it that some individuals appear to be asymptomatic, or they appear to be immune, or they appear to have very mild symptoms, or never get infected by a particular pathogen, we might actually, if we understand that, we might find out or have ideas, have new insights about how the disease takes place, and to develop new preventive strategies. So that's why we call this the remedy within, or the drugs within. So exploring our own human body for finding new strategies to combat disease. In particular, in this talk today, I'm going to focus mainly about harvesting our own microbiome for finding new prebiotics and health associated bacteria. So that is also probiotics to prevent oral diseases and gut diseases and so on. And but in the lab, we have been also working about immune system, different antimicrobial peptides, which are part of the innate immune system in humans, which also appear to be specific or at very high levels in individuals which appear to be immune. So today, I'm going to be focusing mainly on the human microbiome. You know that it is this set of microorganisms, both fungi and bacteria, viruses, and so on, that appear to live inside our body under normal conditions. And I think this has really shifted our attention a lot from, thinking in general that microbes are the source of disease, to actually looking at, many microbes in our body that actually are fundamental, are instrumental for our health. And we have these bacteria, fungi, and so on in the gut, which is the most common, the one that has been studied the most, but also in the oral cavity, the respiratory tract, in the skin, and so on. So that a big portion of our body is formed by these bacteria. The estimated number of bacteria is more or less the same. Some people say that even higher than the number of human cells, and they encode about 100 times higher, between 100 and 1,000 times the number of genes in our own human genome. So imagine, when we look ourselves in the mirror, we have to think that we are half microbes, half human, and they actually can perform incredible set of functions for our health that only now we are starting to understand. So that's why I call this I know that this term doesn't like, not everybody likes it, but I like to consider the human microbiome as a human organ, of course, from the physical point of view, but it is, in my opinion, from the functional point of view, because it's able to perform some functions that we cannot do without them. And this is, for example, because we don't have the appropriate enzymes, and our bacteria do, and therefore they provide some functions that we cannot do. An example of this is provided by some bacteria that we have in the oral cavity, which are able to transform the nitrates from the vegetables, NO3, into nitrite, NO2, and then that nitrite, NO2, into nitric oxide. That second step can be performed by our own enzymes, but this first step, we don't have the enzymes to do that, and we rely entirely on these oral bacteria to do it. Nitrate actually comes, we ingest more than 80% of the nitrate from the vegetables and fruits. So it's very common in green, leafy vegetables, and also from other vegetables like beetroot and so on. There's also a little bit on fruits. And what happens is that when we transform this nitrate into nitrite and then into nitric oxide has many important functions in the human body. For example, this is a very important vasodilator, which means that when we ingest vegetables, our blood pressure can go down. If you tell me 10 years ago that oral bacteria were able to have an influence on our blood pressure, I would have not believed it. And this is one of the examples that really show in a beautiful way how they can perform functions that we cannot do. So that's why I say that it's like an organ. And actually, when we began to work on this, we thought, okay, how long are going to be these oral bacteria in the mouth in contact with the vegetables, to perform this function? It's just going to be for a few minutes while we are chewing on the food. Most of the nitrate is going to be swallowed, and it's probably going to be lost in the urine, And this is when things start to get really interesting, because although part of the nitrate is lost, a big part of it is going to be recycled. It goes to the bloodstream. And when the blood goes near the oral cavity, the salivary glands actively capture the nitrate and put it back into saliva, which is incredible. So the human body makes sure, it ensures, our evolution ensures that we recycle and we use the nitrate. We provide the nitrate to that mini factory of bacteria that we have in the oral cavity to perform this function. So, which is, in my opinion, one of the most beautiful examples of how the human microbiome is highly integrated into human physiology. So this is the cardiovascular effect that we see in the lab. The way we test it is that we, for example, ingest a glass of beetroot juice, which is very rich in nitrate. It could be lettuce juice or any other vegetable which has a lot of nitrate. And then we swallow it. We measure the level of nitrate in saliva, and we see that after half an hour, we, like a miracle, we finally see this peak in nitrate in the oral cavity because it has been already recycled and captured by the salivary glands. Then we swallow it again, it gets recycled again, and by doing that, our body keeps the nitrate in the oral cavity for about 6 hours. We see that this varies between individuals, and we still have to see what's the effect of this for blood pressure and cardiovascular events. The nitrate gets transformed into nitrite, and the nitrite, either by the bacteria or by our own enzymes, into nitric oxide, and this nitric oxide performs this vasodilation of the blood vessels. It improves endothelial function, and it has many positive effects, ranging from better sports performance to an improvement in cardiovascular health. So when we measure actually the nitrite, NO2, in saliva, we see that there is again a peak, like half an hour later. So this nitrite comes from the reduction of nitrate into nitrite by the oral bacteria. However, if we have a mouthwash with an antiseptic, that kills all the bacteria for a while in our oral cavity, and we measure the nitrite, there is none. So all the nitrite that comes from the nitrate has to be produced by oral bacteria. So this is why, in my opinion, having a mouthwash on a daily basis is not good for your health, because among other things, is going to reduce the availability of nitric oxide in your body, because you are killing not only the bad bacteria in the oral cavity, but also the good ones, which are able to use the nitrate that we cannot do. So this is very interesting because we could see a positive effect of oral bacteria. And we see that when we have disease, for example, when we have gum inflammation or gum disease, then these bacteria go away. So, for example, and this happens in different samples around the world. So when we look at, for example, the levels of pathogens in people with gum disease compared to healthy people, we see that there is an increase in all these pathogens, and it is the other way around for the nitrate-reducing bacteria. So we increase the pathogens and we lose the bacteria which reduce the nitrate. And that means that if you have gum disease or bad oral health, you lose these bacteria and you are going to have lower levels of nitric oxide, and you are going to have higher risk of having, high blood pressure and many other complications which are derived from having low nitric oxide availability. So this is very interesting, and this was done in collaboration with a team in Mexico. Nidia Sanchez, who is doing her PhD under the supervision of Professor Lucrecia Carrera, who is also a good friend. And what they did there was actually to prepare beetroot juice from a fresh beetroot, and then gave it to healthy people and people with gum disease. And both the systolic and the diastolic blood pressure actually goes down 2 hours after drinking this beetroot juice. So it goes down in healthy individuals. However, when you have the disease, when you have gum inflammation, this gum disease, there is no change in blood pressure, because you don't have these positive bacteria that are able to transform the nitrate into the nitric oxide. However, what they did is that with the help of a dental clinic, they treated these individuals. So their oral cavity, their gum microbiome, the healthy gum was restored, and these bacteria came back to the oral cavity again. And when after 70 days after the treatment, when they had again this beetroot juice, then again they had an improvement in blood pressure. So this is a very neat, in my opinion, the demonstration, experimental and clinical evidence that actually, having nitrate really improves your cardiovascular health, and you need to have a good oral health for doing that. So that means that we can, have products in the market like chewing tablets based on the vegetables, which actually could improve our oral and systemic health. Of course, we could also improve our diet, eating more vegetables, but it's very difficult in general to change the habits, the daily habits of people, and it's easier. People prefer to, take a tablet rather than go for a walk or do sports, So anyway, in addition to changing your diet, you could also take these tablets based on vegetable extracts. And in the lab, in addition to having these systemic effects, when we apply nitrate, we study these oral bacteria after 5 hours, they have removed most of the nitrate. By 9 hours, all the nitrate is gone. And then when we measure the nitrite, this NO2, we see a peak at 5 hours. And by 9 hours, it's also gone. This is because this nitrite has been transformed, as I mentioned, into nitric oxide and also into ammonia. Ammonia buffers the pH. So when we have an acidic pH, because we eat sugars, for example, that acid in the oral cavity causes dental caries, causes tooth decay. And so eating these vegetables, which are rich in nitrate, they could act as a prebiotic, that is a substance that we ingest in the diet that is able to promote the growth of beneficial bacteria or to perform some positive actions of these bacteria. In this case, the production of ammonia that buffers the pH, and that's why eating vegetables is good to prevent cavities. And then, of course, as I mentioned before, nitric oxide is not only good at the systemic level, but also in the oral cavity, it kills many pathogens, because many of the bacteria causing dental caries and also causing bad breath and causing gum disease, gum inflammation, like the ones which are here marked with a red dot, which is called the red complex of periodontal bacteria, they also are decreased in the presence of nitrate. And then there are some bacteria which are associated with health at increasing proportion. So that's why we believe that adding a source of nitrates to toothpaste or to any products, could be big advancement. In addition to killing this harmful bacteria in the oral cavity, we have seen also, especially at very high doses of nitrate, that there is a decrease in the formation of the biofilm in the oral cavity. So I anticipate that in a very recent, in a very near future, we are going to see nitrate-based or vegetable extracts-based toothpaste. We are already in collaboration with several companies to develop this, and I really anticipate that we are going to see this. And I am very excited about this because from looking at work that we have done in the lab, we believe that we can really improve oral and systemic health by the introduction of this product. And of course, not only we could have these tablets rich in nitrate-rich vegetables, but also we could try to incorporate these bacteria, which are able to do this job, in the mouth of people that do not have them. And it's very interesting because we have developed these agar plates with a chemical reagent that turns red in the presence of nitrite. This was, basic chemistry. It's this is called the Gries reagent. And what we do is that we put the agar plates with nitrate, and then we culture bacteria from healthy individuals. And those bacteria that are able to transform the nitrate into nitrite, they appear with a red color around. So we can actually pick those colonies and then try to develop these bacteria as health-associated bacteria or probiotics, that we can try to incorporate in the oral cavity of people that have low levels of those. But what happens if we cannot culture those bacteria? Then we have a trick, which is to construct metagenomic libraries. What we do here is that we get DNA from the oral cavity or from the gut or from any environment with bacteria that we cannot culture, and then we fragment that DNA into little pieces, and then we clone it. That is, we insert those DNA into a bacteria that we can culture in the lab, like E. coli. So now we have, we repeat these thousands and thousands of times, and have thousands of strains of E. coli, each of them with a piece of DNA from bacteria that we cannot culture. So now we have those inserts of DNA into the bacterium. What we can do is that we, in those plasmids that contain the DNA, we induce them to multiple copy. And then we have E. coli, which is going to produce whatever is encoded in those DNA inserts. So then we can make some function-driven analysis where we can actually check which proteins or which compounds can be synthesized by that DNA from those bacteria. So we wanted to look for compounds to kill oral pathogens. And what we did was to repeat these, 20,000 times, where we cloned DNA from the oral cavity of people that had dental caries. And this is more important for us, people that never had a cavity in their life, these caries-free individuals. This is about 10% of the population that never had a cavity. Sometimes they even say, no, I never brush my teeth and I never had cavities. So we wanted to know why these people appear to be, immune to cavities. You can see here, each of these colonies here is a strain of E. coli. They are all identical, so they are all the same clone of E. coli, but each of them has a different insert of DNA that comes from another bacteria. You can already see that even the shape of these colonies is different because these bacteria are expressing, the DNA which was inside and comes from another bacteria. So what we did was to test those 20,000 strains of E. coli against the pathogens of dental caries. We have here lone culture of the bacterium causing dental caries. And here, these colonies here are the different strains of E. coli that gave a positive result. As you can see, there is an inhibition ring around it, which means that the DNA that we managed to insert there was being expressed, and whatever compounds were encoded there, they were diffusing in the agar and were preventing the growth of the pathogen. It was extremely exciting to see this, because then what we did was to sequence the DNA, and then we identified DNA from, similar to the DNA of some bacteria, which appear to have antimicrobial peptides that we are now trying to develop to kill some oral pathogens. It was very interesting, and actually we had a very big surprise that I can I don't have time to go through that today, but actually we had another 6 of these DNA inserts that were not bacterial, they were human, actually. So we had pieces of human DNA inserted in E. coli that were being expressed and were killing oral pathogens. And we're like, what is this? And what happens there is that we were just by chance inserting pieces of DNA containing encoding the genes for antimicrobial peptides from our human innate immune system. And by that way, we identified new antimicrobial peptides that kill some of these bacteria. So just to, in one slide, I'm just going to tell you, so you know, perhaps we can talk about this in the near future, that by doing this, I'm convinced that the human genome is full of these antimicrobial peptides against viruses, bacteria, fungi, and so on, that we don't know. We only know a few dozens of them, but by doing this, we just by chance, we happen to clone DNA that contain this antimicrobial activity. So we decided to do the same, not against dental caries, but what happens with people that never catch the flu? Even in a family, we have people that some of them get the flu and others do not. And these asymptomatic individuals that appear to be immune to influenza, what we did was to collect samples from those, and then we saw that they had, high levels of an antimicrobial peptide that was produced. We found it to be produced in saliva, in the respiratory tract, even in the tears. So it was actually blocking the entry of influenza. So I will, this is out a little bit of the general topic of finding new agents in the human microbiome, but this strategy of looking at healthy individuals, individuals apparently immune or resistant to the infection, can be taken to other diseases. And we apply the same principle, and found a human encoded antiviral peptide that appears to block the binding of influenza to the human cells. And we have now done animal tests showing that it actually works. And we are now trying to find a private company to develop this as a drug against influenza, which would be wonderful. Okay, and the last story I wanted to talk to you about is it actually all started with this, whether a keys can actually be the key to understand why some people were immune to dental caries. And so this is why Dr. Ramy was saying before, whether, can marriage save your teeth? All this came because there is, as you know, about 10% of the population that never had cavities. And one of these person was in our lab when I was studying, when I was doing my postdoc. And we were having lunch one day and she said, I've never had cavities, but in my life, but my boyfriend used to have lots of cavities before going out with me. But when we started going out, then he stopped having cavities. So that's when we thought that perhaps she was passing him some of her good microbes in the oral cavity that might have a positive effect preventing dental caries. Of course, I asked her, "Are you sure that perhaps your boyfriend didn't brush his teeth before meeting you?" But and now he does. But she insisted that wasn't the case, and so we began to work on that. So we began to look at the bacteria present in caries-free individuals. So this was the first metagenome that was published worldwide, which is sequencing the total DNA of the bacteria inhabiting the oral cavities. We collected dental plaque samples from caries-free and caries-active individuals, and then we sequenced little pieces of DNA of all the bacteria present there. So we could know which bacteria are present in those individuals, those lucky individuals, and what is the function that they encode. And we identified that in the individuals that never have a cavity, they have high levels of these two bacteria, Rothia and Neisseria, that we didn't understand at the time why they were at very high levels. Now we know, because these are the ones that are able to utilize the nitrate. So now I'm very happy, because after these years, we closed the circle, and now we understand why. They use the nitrate, they produce ammonia to buffer the pH, they produce nitric oxide that kills pathogens. And in addition to that, we also found very high levels of what appeared to be a new species of Streptococcus. So we compared to the Streptococci that were present in databases, and this was a different one. So we said, okay, this one we can culture. So let's go to the samples of this girl, that never had cavities, and others, caries-free individuals. And then we took samples and were trying to capture, to culture and to find these Streptococcus associated with good oral health. We managed, and it was indeed a new species. So it was very interesting very exciting to give a name to this new species for science that we call the Streptococcus dentisani, which means healthy teeth in Latin. And it is this beautiful bacterium here. And we wanted to know why it was, associated to health. At the beginning, we thought that perhaps it was able to grow faster than the pathogens and it would occupy the teeth or something. But actually, when we saw this, it was one of the most exciting moments in my career. What we have here is a culture of Streptococcus mutans, one of the bacteria causing dental caries. Here, Sobrinus, another bacterium causing dental caries. And what we see here is this Streptococcus dentisani. And as you can see, around the colony of dentisani, there is an inhibition ring because dentisani produce many bacteriocins, many antimicrobial peptides that inhibit the growth of the pathogen. Here we have another bacterium that doesn't produce any. Here we have another one that produces a little bit of inhibition. So we selected the ones that were most active against the pathogens. It was incredible because then we understood, that people without cavities have good bacteria that actually prevent this from happening. And now this idea is very clear and is well established. But when we first wanted to do research on this, actually we didn't even get any funding. The first experiment I did it paying it with my credit card. So I mean, I didn't get the national grant to study this in the first place. So it was a very exciting moment. And actually I have captured, I have rescued an SMS, a message that I got in my old Nokia phone a long time ago from the student that did this experiment, because there was not even WhatsApp yet. So he sent me this message on a Sunday because he put the experiment on Friday and he couldn't wait until Monday to see the results. So by Sunday, he goes to the lab and then he sees that there is inhibition and he got so nervous that he sent me this message that I didn't understand the word because he made a lot of errors when he was typing. And he said, "Hey, Alex, that the inhibition paths seems there is something. I see rings. Well, I don't know. We see on Monday." So he was so nervous that it didn't make sense at all. I mean, he meant that there were inhibition rings in the agar plates, and he thought that there was something there. Anyway, so I phoned him, and then we were talking. It was a very exciting moment, Of course, things in the real life, it never happens like in the Hollywood movies, but this is the way that it happened. We were very excited about it. Then the whole thing is expanded. We thought, okay, let's use these bacteria to put them into a yogurt or a toothpaste or a chewing tablet or something. We began to check around the world, including also in Egypt. I'm very grateful for the samples that you sent me from there. And we saw that caries-free individuals without caries had very high levels of dentisani, and individuals with caries, they have very low levels or even absent levels. So we thought that this was a good idea, try to develop this as a probiotic. And then we got also a lot of people that contacted me in relation to the kissing. They wanted to test whether kissing each other were going to pass bacteria from one individual to another, and to what extent this could be the origin, the evolutionary origin of kissing anyway. I thought that was going to be terrible. So I said, no, I'm not going to go into that. But then actually a team in the Netherlands did exactly that with the students. So they got the students to have control what they called intimate kissing for 10 seconds. Then they measured the saliva before and after the kiss to see whether there were bacteria that were passed from one to another. And actually they found that about 80 million bacteria were passed by these 10-second kisses, which was incredible. And they began also to look at, what happens to the bacteria, how many bacteria you share with your partner, depending on how many kisses per week you give to each other. So you give a lot of kisses, more than 120, then, your bacteria in your oral cavity are actually very similar. And if you don't kiss much, then they are, very different. And it is the same here, the longer time that has passed in hours since the last kiss, it means that you are losing bacteria in common, and then the composition of bacteria in the saliva from those two partners is different. So anyway, then we wanted to know how dentisani is killing S. mutans and the oral pathogens. This is an EM picture of S. mutans, one of the caries pathogens, after being exposed for 10 minutes to the peptides produced by dentisani. And I'm going to mark here with my mouse, where there are little holes. You see here, here, here. These are pores, which is the typical here, all these pores are the typical mode of action of antimicrobial peptides, which normally have a positive net charge. So they bind to the cell wall of bacteria, which is negatively charged. They are normally hydrophobic, so they form these pores in the lipid membrane, and then they make holes. And there is a point that the bacteria has so many holes that it's not able to keep the osmotic pressure between the inside and the outside of the cell. And what happens then is that the bacteria lyses. And this is where we see, how these are the remnants of S mutants that has they are cocci, so they are round. And here, all the internal components have been released outside because it has been lysed by the bacteriocins of Streptococcus dentisani. So this is how bacteria kill each other. And this is what we believe is happening at the microscopic level in the dental plaque of these caries-free individuals. And we wanted to understand which molecule was being produced. So we sequenced the genome, and then we looked for these antimicrobial peptides. And then we found actually the largest number of bacteriocins in any bacteria known so far. So it has 14. So it's not only one, it has not 2, not 3, 14 of these antimicrobial peptides. In bacteria, they are called bacteriocins, and they are very small peptides between 25 and 50 amino acids. And most of them appear to be in an island here, in a cluster in the genome that we have called anti-caries defense cluster. The reason for this is because I like the ACDC, the band, so we decided to keep this name, and no one said anything in the journal, so now the name is there, anti-caries defense cluster, the ACDC cluster, where most of these genes encoding the bacteriocins are present. So this is the list of the 14 bacteriocins that we have called denticins. Some of them were very similar and known, so they were known already. Sorry? Because they were present in other bacteria, but most of them were new to science, and we call them denticins, from denticin A to denticin H, and we are now looking at what's their structure and which bacteria they are active against. So now we have two options: to develop the bacterium as a probiotic, or to use the denticins to combine the denticins in a cocktail that targets specific oral pathogens. After our discovery, there has been the development of other bacteria which appear to be associated with health, like the Streptococcus A12 in Florida in the US, which appears to produce a lot of ammonia and therefore is able to buffer the pH very well, or others. The way that we have applied it in dental clinics so far in the clinical trials is in this gel, this bucco-adhesive gel that we apply for 5 minutes. And we have seen that it actually improves the pH, it lowers the level of pathogens, it lowers gingival inflammation, and so on. So we are very close. Actually, it is going to come out to the market at the beginning of next year. In Europe, it's going to come out probably in toothpaste or in a product for professionals. In the US, it's probably going to be as a chewing tablet. I don't know what it's going to be like in Egypt, but we'll see. This is very exciting because after all these years, I have seen all the process from discovering something in the lab that you believe is interesting to actually develop all the in vitro testing, the animal safety trials, the human clinical trials, the scale up, to be able to produce it at a high scale, being low price enough to be able to be viable and being able to take into the market. And it has been, I'm telling you all these steps so you can see actually that it takes a long time to develop a product, but I think it really pays off. And after the Dentisani, all the research in my lab has been applied, and we are working on the development of new products for improvement of oral health. So the Dentisani will come out at the beginning of next year to market, and the idea is to have two types of product: one for the professionals that the dentist can apply in the dental clinic with a varnish or a gel at high dose. It will be a bit more expensive with high doses, and this could be like a shock treatment. And then the Dentisani for the general use that could go into toothpaste or tablets and so on, like for a maintenance treatment that will go through, that will be at lower cost. So I think this is a nice example about how basic science can be translated into the real world. And I think, we all knew that from the point of view of love, of course, kissing is good for your health. But now I think that science can actually, it has been able to confirm this, that actually can also improve your health, at least your oral health, especially if you kiss the right person. So I want to thank my team. It has changed a lot from the beginning of this project until now. And also the people that have worked with me at the University of Jönköping and at the University of Guadalajara in Mexico. And also especially in Valencia, Carmen Jena at the University and Dental Clinic for helping us in all the development of the clinical trials. So thank you very much for your attention and for the invitation, and I'm happy to take any questions if you want.
Thank you very much, Alex. A very, very interesting story, of course. And we don't need really in the technology age to open the floor for questions. You already have at least 7 or 8 questions in the chat and in the Q&A. There are two signs here, chat and Q&A. Both of them are good for questions. But before opening the floor, I will just ask you, first of all, thank you so much for the very interesting story. And thanks also to all more than 70 attendees at this evening hour. So it's evening in Spain, it's evening in Cairo. So thank you all for being here. So let me just open the debate or the questions with a couple of simple questions, and then you can feel free to browse the questions and choose which one you want to answer, or if you want me to read them for you, that's fine. So my simple question is, so you're not a very, from what I understood in the first part, you're not very positive about using mouthwash. You think mouthwash may have some problems. And the second question is, well, it may sound like a joke, but it's a real question. Do you think that we should add microbiome screening to premarital tests?
Okay, in relation to your first, well, I mean, I will start with the last one. Yeah, I mean, I think it is funny because it is true that there might be some incompatibility, between the microbiome of different people, but but anyway, I think that once you cohabit the same house, you start to share also a lot of your bacteria, mainly the one in the oral cavity, which is the one which is most exposed, but also the one in the skin and so on. This has been shown several times. And in relation to the mouthwash, of course, I think it's good to use the mouthwash for specific purposes. So, for example, if you have a very bad gum disease or a lot of inflammation, or you have had a tooth extracted and you want to have extra care to prevent infection in the bloodstream, I mean, for specific periods of time, according to what the dentists say, I think it's totally fine. What I don't think is a good idea is actually to sterilize your mouth on a daily basis as part of your oral hygiene routine, in the same way that we wouldn't drink antiseptics every day to sterilize our gut flora, because we know that they perform very important functions, and the same is true for the oral cavities. I think it's okay for particular situations, but not on a daily basis. And I'm seeing here some questions.
Sure. While you can check and read some questions, and may I first ask if any of the panelists, to our guest of honor, Professor Khayyal or Dr. George, if the panelists have a comment or a question, and then while Alex is reading.
Thank you, Ramy. I would like to thank Dr. Alex very much. I enjoyed the lecture very, very much.
Thank you.
Because it means to me, to do good research, you have to let your teeth into it. And then I've learned also a lot that to have a kiss, it's better to have a kiss in the morning than a kiss at night. And you know why? Because at night you have a nitrate. No, I really enjoyed it very much. And muchas gracias. I really enjoyed it very, very much. Thank you so much.
Thank you very much,
Studios,
Dr. Alex, for such interesting story you told us today. And it's really interesting and having good advantage for marriage and kissing. Today we learned a lot about kissing and marriage. So we enjoyed, and I'm leaving Dr. Ramy to read some questions to you.
Yes,
I think we are having a bunch of interesting questions.
Yes, and note that a lot of attendees are also from MARC or Eva Pharma, so maybe we can think about taking some rights and having the first Egyptian product soon. So the very first question that was sent in the QA session was by Mina Abadir, and he talked about his experience, but his question to both speakers is, how can pharma companies come on board with natural wellness keys such as the microbiome to cure and prevent many diseases instead of treating just symptoms? So basically the question is how feasible this, I mean, your story is one of those beautiful stories that doesn't happen every career, but in general, is it feasible? Is it doable that pharma companies can start looking at natural wellness keys? The main question is, can we focus on health rather than treating symptoms? I don't know, Dr. Khayyal or Dr. Mira, if you have any comments on this.
Well, I have one thing for sure. Number one, we don't have proper microbiology research in our faculty. And it's very important, to develop the microbiome. Nowadays, I find when I'm talking about herbal medicine, without the microbiome, I'm lost. But there's not enough research work going on in Egypt on the microbiome. And most of the research work at the moment is being carried out in Europe, in Germany, in Austria, and everywhere else. But we have to develop research on a better standard. And at the moment, I find it lacking. I would love to see more on the microbiome. My work on herbal medicine has been now changed into microbiome, and I did my best to do some work on the microbiome with the herbal medicines I'm dealing with, but there's a lot to be done. But who is really interested in research? I find it difficult to find the right people for the right job. I'm sorry, but
no, we are trying. We are trying.
I know.
So Alex,
I am a born optimist, and therefore have to keep on trying, never lose hope, and inshallah. Lovely, very interesting talk. Thank you.
Yeah, Dr. Alex, any comments on this question, or we?
Yeah, well, two big things. One, I of course, I do believe that the pharma industry has to be evolved. I think that many of the discoveries found in academia are never translated into a real product because there is a very big lag, between academia and the real world. And I think there has to be higher chances for interconnecting those two worlds. I think it's very important because with public funding, you can make the discovery and perhaps do some proof of concept in an animal trial or in animal test. But if you really want to take this further, you need a very big investment, which is normally not covered by the science funding agency. So you need them to be involved. And in relation to what the question said about treating not the symptoms, but actually the source of the disease, I think this is a very important point because in many occasions, we take for granted that the disease is going to be impossible to be prevented. It's like, for example, with cavities, we assume that they are impossible to be prevented. And then there is a lot of research, if you go to a dental congress, about fillings, how to film or how new kind of materials for the fillings or for having new teeth implanted and things like that. It's as if the job of us as clinicians would start once the damage is already done, once the hole is already done. But can we actually think about how that is, going to be produced, and are we able to prevent it? And that's why I think it's interesting to look at people that don't have the disease. We found the same with the flu. Now the current treatments, the main current treatments for the flu actually treat the symptoms. You feel bad, and then you take something for the headache and so on. But what we are seeing here is like, okay, this goes by influenza. Can we block the infection? And that's why, by looking at these people, asymptomatic people, then we were able to develop, to find this antiviral peptide that could be perhaps used as to put into a product, into the nose, in a spray into the nose and into the mouth, and it could actually prevent or treat the disease as opposed to treating the symptoms. So I do agree with this question, and I think it's something that we need to take more into account, and not assuming that it is going to be impossible and to start our job once the disease is there, by trying to prevent it. I think it has to be possible in many cases.
Great, thank you so much. One of the early questions too were from Dr. Hadi Kyriakos. It's a clarification question. Were arterial nitric oxide levels measured in people with periodontitis?
Okay, to measure nitric oxide is very difficult because nitric oxide has a very short half-life. So after a few seconds, it is gone because it gets normally oxidized into nitrite. So you can measure nitrite, and yes, of course, it has measured, and the nitrite levels in blood in people with gum disease are affected. And actually, there are a lot of links between having bad oral health, especially gum disease, and having a lot of systemic conditions, because it influences obesity, cardiovascular health, the diabetes, and many different things. And the general idea is that oral bacteria, oral pathogens create a systemic inflammation state, by the production of many pro-inflammatory molecules and so on. And now we have seen that in addition to that, part of the effect is due to the lack of the good bacteria. By not having these health-associated bacteria, in people with gum disease, then you don't produce the nitric oxide. So I think it's not only what you get when you have the pathogens, but what you stop having when you don't have the good bacteria. I'm also seeing.
Okay. So you can see many questions. So maybe you can pick your question, because we have about 6-7 more minutes to answer. So you can pick the questions. There are technical questions about how to sample buccal swab or not. And there is a question also, are these products in the market already, or they are approved, registered? And then there is a question on oral cancer and smoking. But again, you check the chat.
Okay, thank you. Well, the Dentisani and the nitrate, they are both not in the market yet. The Dentisani will come out to the market next year, although I don't know in which regions. I think it will be Europe first, then the US, and then the rest of the world. In relation to the nitrate, the company that is going to put that into a toothpaste has decided to have some more clinical evidence before taking it to the market, because they have to decide the dose and so on, and they want to have that right. So we are going to start now the clinical studies, and it will take 2 years to be in the market. Yeah, and then the other question here, from where the human genes inserted in the E. coli with the antimicrobial activity are coming? Was it contamination or okay, I see. Yeah, what we did was to get dental plaque samples, which is all the bacteria that accumulate on the teeth. But of course, there, in addition to the bacteria, you also have human buccal epithelial cells and immune cells, and these buccal cells that detach from the buccal epithelium, and then they end up being attached to the dental plaque as well. So when we extracted the DNA of the microbiome, about 10-20% of the DNA was actually human, and that human DNA happened to be inserted also in the plasmids, and that's why we saw activity coming from human DNA. So it wasn't a contamination in a way, because our sample already contained human cells and human DNA.
Regarding buccal swabs, or is it proper? I mean, just technically, what is the best sampling way for an oral microbiome study? I think that's the question.
Okay, I see. I think it really depends on what is the purpose of your research. We are very lucky because we have access to the different niches and the different sites. This is very different from what happens in the gut. In the gut, most of the research is based on fecal samples. And of course, a fecal sample is whatever comes through the whole, through the whole gastrointestinal tract. And perhaps some of the diseases you are looking at actually happens in the small intestine. And therefore, when you take a fecal sample, only a small portion of bacteria come from the small intestine, and therefore we're going to have a lot of noise. However, in the oral cavity, you can take the sample from the site. So if you want to study dental caries, then I think you have to take a dental plaque sample, what we call a supragingival dental plaque, from the surface of the teeth. If you want to study gum disease, then you have to take the sample from inside the pockets, the gingival sulcus in the gingiva. Or if you want just to have a general idea of all the bacteria present in the oral cavity, then it is better to use a sample of saliva, because saliva bathes the whole tissues in the oral cavity. And then if you collect a non-stimulated saliva sample, you're going to have bacteria from the palate, the tongue, the teeth, the gingiva. So probably a saliva would be better if you just want to have a general idea. But if you want to focus on a particular disease, choose the sample from the site where the disease takes place.
Very good. There are still more questions coming, and we have like 3-4 minutes. The last question I see is about. So I don't know if you're a proponent of using Streptococcus dentisani, the whole bacterium, as a probiotic, but if you do so, the question is, how do you see its protective effect in individuals with chronic conditions such as diabetes, with all the subsequent dysbiosis? And could this diminish its probiotic activity? Can we target it? Long question.
Yeah, well, it's difficult to know. I think that so far the clinical trials have been done on individuals which were systemically healthy, and individuals with caries and individuals with gum disease. But so we really don't know if the degree of dysbiosis in individuals with other systemic condition could have an effect. What we have seen is that actually, even in healthy individuals, the degree of colonization by S. dentisani is not that great. I think this is a problem in general in probiotics, and it seems that once we are adults, we are already, our immune system is already recognizing the bacteria that has lived with us for all of our lives. And actually, this can be easily seen when we take antibiotics. We recover both in the gut and in the oral cavity. We tend to recover about 1 month or 1.5 months after taking antibiotics. We recover most of the bacteria that we had before. So it's like our body already recognizes them, and then they come back to occupy the niche. That means that the same thing happens when we apply a probiotic. We apply it at high doses, and then it's able to colonize for a few weeks. But after a couple of months, they start to get lost, and we go back to the bacteria that we had before. So this means that we might need to get these every few months if we want the effect to be preserved. Actually, in the clinical trials, it was very nice because even though we saw that after the end of the S. dentisani application, the levels of dentisani started to go down, the improvement in the clinical features, like the improvement in pH and things like that, and gingival inflammation, they kept improving through time, at least for 2 weeks or 4 weeks after the last application of the product. So this means that what probably is happening is that S. dentisani improves the dysbiosis state to having lower levels of pathogens, higher levels of health-associated bacteria, and even if the dentisani levels go down, at least for some time, the clinical improvement still holds. So we still don't know how often this would need to be applied.
Great. There are still more questions coming, and I think we have everything recorded and saved, so Alex can also correspond with the questions. But there was a question that has been submitted a while ago, so maybe you can take it as a last question by Ahmed Sharawi. High incidence of oral cancer has been linked to tobacco smoking, and claims that NO, nitrous oxide, plays a part for either initiation or progression of carcinogenesis. So it's about the role of nitric oxide in carcinogenesis. Does it have a negative role?
Okay, two things. One is the nitrate. I mean, the source of the nitrate is very important. So, for example, if you get a nitrate as a preservative in added foods, like in ham, sausages, and so on, there you add a salt. And then when you add a nitrate salt, actually that nitrate is transformed by the bacteria in the meat into N-nitroso compounds, and amines and I mean many different products which are known to be carcinogenic. However, when the nitrate is not as a salt, but actually comes from the fruits and the vegetables, there it is combined with antioxidants, polyphenols, and so on, which actually ensures that that nitrate is going to nitrate and nitric oxide and not into the compounds which are harmful. And also in relation to the question, nitric oxide is very important for our immune cells to kill cancer cells. So actually improving the nitric oxide bioavailability is known to also improve the fight against cancer. So this is one of the reasons, along many others, why having a diet which is rich, which is very high in in vegetables, including green leafy vegetables which are rich in nitrate, there is always a correlation with the ingestion of these vegetables and the lower cancer rates. So I think that has been shown over and over, and I think part of the confusion comes from the source. We have just published a paper which is called "Nitrate: The Source Makes the Poison," as opposed to the dose makes the poison, because if the source is fruits and vegetables, then it is very positive for your health, including lowering the risk of cancer. But if the source is actually a nitrate salt, which is added to ham and so on, and bacon and so on, then that's bad for your health.
Dr. Alex, Mira, thank you so much. So many questions. The more you answer, the more questions come from the audience. So of course, we will make sure. Again, Alex Mira, you can search his name. Fine. There was a question about the paper, so you can also search the publications. So thank you so much for this. We would have loved to stay also longer. I know that people in Spain love football and love staying late at night, but maybe another time. And Professor Khayyal, thank you. Yeah, this year Valencia is not doing very well, so let's not talk about it.
Oh yes, I know.
Most Egyptians support either Barcelona or Real Madrid, so
I know.
And Professor Khayyal, thank you so much for staying with us till the end. We really appreciate the good example that you gave so many generations of us. And again, there are so many questions, and I believe that, so again, for the questions about this presentation, like the past webinar, we take a little time to process it and make sure like there are no extra parts. So it takes about a week editing, and it will be on MARC website on YouTube, MARC YouTube, and then it will be shared on social media. So thank you so much. It was really enjoyable. And although I have met Alex a few times and listened to his talks, half of this talk is all new to me. Professor Khayyal, I always learn new things from you. So thank you so much. Dr. George Maher, please, you can give the final word.
Thank you, Dr. Ramy. Thank you, Dr. Alex, so much for such interesting talk today. And I'm inviting you, if you will pass by Egypt anytime, to visit us in MARC. It will be having our honor to have you in our MARC site. And I'm sure that we can find a collaboration together in your research, and I would love to have such products you talked about today by MARC and Eva Pharma to the Egyptian market if there is any chance to have such collaboration. Thank you, Dr. Khayyal, for being with us, and it was really enjoying for us all, and we enjoyed having spent this night with you both today.
Thank you, Dr. George. Thank you, everybody. And by the way, Professor, Dr. Alex has helped so many in the department already with so many ways, and he's in a few of our publications in the Department of Microbiology. So we look for more. Thank you so much, Professor Khayyal. We are also awaiting your visit, inshallah. And with this, good rest of the night to everybody. And look for next webinar, inshallah, on October 28th.
Inshallah. Okay. Thank you. Bye-bye.
Thank you. Bye-bye. Thanks a lot.
Bye-bye.